Welcome to our exciting journey into the microscopic world of cell biology. Today, we're going to explore one of the most fascinating and absolutely essential molecular machines in existence, known as ATP synthase. Get ready to discover the tiny biological engine that literally powers almost every living thing on Earth. Now that we know what we are looking at, let us understand how it works through a simple analogy. Imagine a massive hydroelectric dam built across a powerful river. The dam holds back water, creating immense pressure. When the gates open, water rushes through, spinning giant turbines to generate electricity. In your body, a nearly identical process happens trillions of times every second. Instead of a concrete dam, you have a biological membrane. Instead of water, you have tiny positively charged particles called protons. And the magnificent spinning turbine is ATP synthase, generating the energy currency your body needs. Building on that analogy, let us look at the actual structure of this biological turbine. ATP synthase is a highly complex, multi-subunit enzyme embedded right in the inner mitochondrial membrane of eukaryotic cells. It is divided into two main structural domains, known as the F0 and F1 portions. The F0 region is a water-insoluble channel embedded directly inside the membrane, acting as the pathway for protons to flow. The F1 region is a water-soluble, spherical structure that protrudes into the mitochondrial matrix, and this is where the actual catalytic sites for energy synthesis are located. With the structure in mind, let us see this machine in motion. As protons flow down their electrochemical gradient through the F0 channel, they cause a central stalk within the enzyme to physically rotate. This is a literal mechanical motor inside your cells. This rotation forces the F1 subunits to change their three-dimensional shape. As they shift, they forcefully bind adenosine diphosphate, or ADP, and inorganic phosphate together. This mechanical action creates adenosine triphosphate, or ATP. It is a beautiful translation of physical movement into chemical energy. Understanding how it moves brings us to why this movement is so critical. Almost every active process in the human body requires ATP as a fuel source, from blinking your eyes to solving math problems. Without ATP synthase, our cells would have to rely solely on glycolysis, which is a very inefficient process that produces only a tiny fraction of the energy we need. If this enzyme stops working, which can happen in the presence of deadly poisons like cyanide or oligomycin, cellular energy production halts immediately, leading to rapid cell death. It is quite literally the engine of life. Now that we appreciate its importance, we must ask, what exactly pushes the protons through this enzyme? The energy that drives the protons through the F0 motor is called the proton motive force. This force is created by the electron transport chain, which actively pumps protons out of the mitochondrial matrix. This pumping creates both a chemical gradient and an electrical gradient across the membrane. The combination of these two gradients creates a powerful electrochemical pull that forces protons back into the matrix through ATP synthase, spinning the biological turbine. This electrochemical pull we just discussed can be expressed mathematically to understand exactly how much energy is available. The formula shows that the total proton motive force, delta P, is the sum of the electrical charge difference and the chemical concentration difference. Delta psi represents the electrical membrane potential, which is the charge difference across the membrane. R is the universal gas constant, T is absolute temperature, and F is the Faraday constant. Finally, delta pH represents the difference in proton concentration. Physically, if either the charge difference or the concentration difference increases, the total driving force pushing protons through the enzyme increases. Let us apply this formula to a biological scenario to see how changes affect the cell. Suppose the electron transport chain becomes highly active and pumps more protons. This directly increases the electrical charge difference across the inner mitochondrial membrane. Looking at our equation, because the electrical potential is a key component of the total force, an increase here means the total proton motive force must also increase. As a result, the stronger electrochemical pull forces protons through the F0 channel at a higher rate. The physical meaning of this is clear. The ATP synthase motor spins faster, producing more ATP to meet the cell's energy demands. Now that we understand the force driving the motor, let us calculate how much energy it actually makes. To solve this, we use a simple ratio based on the biological mechanism of the enzyme. The formula states that the total number of ATP molecules produced equals the total number of available protons divided by the proton cost per ATP. Conceptually, this is just like knowing you have a certain amount of money and dividing it by the cost of a single item to see how many items you can buy. If the total pool of protons increases, the ATP yield increases proportionally. Let us look at a practical example using this ratio to see how proton flow translates into cellular energy. We are given that a specific mitochondrion has pumped 120 protons into the intermembrane space. We also know from experimental data that it takes exactly 4 protons flowing through ATP synthase to synthesize 1 molecule of ATP. We chose our ratio formula because it perfectly models this biological cost. By substituting our known values, we divide 120 by 4. The final answer is 30. Physically, this means the mechanical rotation of the enzyme will complete enough cycles to generate exactly 30 usable energy molecules, wasting no energy. Having calculated ATP from a random pool of protons, let us now connect this directly to the food you eat. During the breakdown of glucose, your cells produce an electron carrier called NADH. When one molecule of NADH donates its electrons to the electron transport chain, the chain pumps exactly 10 protons across the inner mitochondrial membrane. This gives us a fundamental biological constant. By knowing exactly how many protons come from one NADH molecule, we can trace the exact energy yield from a single molecule of sugar, revealing the true efficiency of cellular respiration. To find out how much energy is stored in that single NADH molecule, we apply our previous strategy to these fundamental constants. The formula for the NADH yield is simply the 10 protons pumped by one NADH molecule divided by the 4 protons required to make one ATP. This specific calculation gives us the famous phosphate to oxygen ratio. Conceptually, it tells us exactly how efficient our cellular power plants are at extracting energy. If the proton cost per ATP were to decrease, the yield from a single NADH molecule would physically increase, making respiration even more efficient. Let us complete this calculation to find the exact energy yield. We are given the 10 protons from NADH and the cost of 4 protons per ATP. We use our yield formula to find the ratio. Dividing 10 by 4 gives us exactly 2.5. In plain English, the physical meaning of this final answer is that the energy extracted from one single NADH molecule is enough to spin the ATP synthase rotor just enough to produce 2.5 ATP molecules. This number is incredibly important because it allows scientists to calculate the total energy yield of an entire glucose molecule. Now that we have calculated its efficiency, let us look at the complete picture of this brilliant machine. In this diagram, you can see the inner mitochondrial membrane separating the intermembrane space at the top from the matrix at the bottom. Notice the high concentration of red protons at the top. The F0 subunit forms the channel through the membrane, and the red arrow shows protons flowing down their gradient. This flow turns the central stalk, indicated by the rotation arrow. Finally, look at the large F1 subunit in the matrix. As the stalk turns, it mechanically forces ADP and inorganic phosphate together, shown by the blue arrows, releasing a newly synthesized ATP molecule. To wrap up our journey, let us review the most important ideas we have covered. First, ATP synthase is a brilliant molecular machine that converts the potential energy of a proton gradient into the chemical energy of ATP. Second, it operates exactly like a microscopic hydroelectric turbine, using the F0 and F1 subunits to mechanically force molecules together. Third, we saw how the proton motive force provides the exact electrochemical push needed to drive this rotation. Finally, we calculated that one NADH molecule yields exactly 2.5 ATP molecules. You now understand the math and mechanics behind the incredible efficiency of cellular respiration.